the invitation to come and talk today. I'm Balasubramanian. I'm one of the genetics doctors from Sheffield. Yeah. So my talk, I've tried to divide it into an introduction, a bit about the basics in genetics, specifically talking about this in OI. Some of the research that's happening in Sheffield, touch upon the recent advances, and hopefully finish off with a summary. So by way of introduction, I don't have to explain to this audience that osteogenesis imperfecta, also commonly referred to as brittle bone disease, is an inherited disorder of bone formation. And as you very well know, there is a broad spectrum of clinical severity ranging from hardly any fractures, a normal adult stature, to plenty of fractures identified in the pregnancy and a lot in between. Some of the extraskeletal features, things that are not confined to the bones, include joint hyperextensibility, skin hyperlaxity, hearing loss, dentinogenesis imperfecta, features in the teeth, etc., etc. So back in 1979, David Sillins and colleagues classified OI into what they thought were 4 distinct clinical subtypes. So as you can see, that's very much a clinical classification, which was then subsequently subdivided into A and B. And as you can see, type 2 was the more lethal, severe form, and type 1 and 3 were the sort of more mild forms, and type 4 was the moderate form. So this was very much a clinical classification when the genetic basis hadn't been identified, and most of the inheritance patterns were thought to be autosomal dominant, with the risk of gonadal mosaicism. And I'll describe all this a bit later on. But then just to point out that over the last 30 years or so, the Sillins classification has expanded exponentially in that if you look at the online Mendelian Inheritance in Man OMIM website, there's at least 18 or 19 different subtypes now. And what's more important is most of the genetic basis of these different types of OI are now known. So basically, osteogenesis imperfecta is a single gene disorder. So there's often variations in one single gene within our genetic material. It's often referred to as a type 1 collagenopathy because it's the type 1 collagen which is the basis of our bones, and that's what's affected in OI. And there's 2 different patterns of inheritance that we know of, autosomal dominant and autosomal recessive. So the million-dollar question is, why does it happen? So over the next few slides, I'm going to try and give you a masterclass in genetics, everything you ever needed to know about genetics. So we start off with the basics. We know that our body is made up of billions of cells, which are the building blocks to our body. And we draw a cell as a big circle with a black blob in the middle, referred to as the nucleus. Now, when you look down the nucleus, you'd see lots of these little squiggly structures, what are referred to as chromosomes. Now, these chromosomes would normally appear quite jumbled up within the nucleus, but what the lab has kindly done is arrange them in neat little pairs for us. So you start with chromosome 1, which is your longest pair, and then you work your way down to chromosome 22, which is your smallest pair, and then we have a pair of sex chromosomes at the end. So girls would have 2 X chromosomes, while boys, as shown in this image, would have an X and a Y chromosome. Now all these chromosomes come in pairs because you inherit 1 copy from mother and 1 copy from father. Now that's what the chromosomes would look like, but if you unravel them, basically what you end up with is a long piece of string of DNA, our DNA code, and on this DNA code sits about 20,000 pairs of genes. Now we're not able to look at each of these individual genes when you look down the microscope. But what these genes are basically doing is providing instructions to our body. So each of these genes directs the synthesis of a specific protein molecule which has a specific function in the body. So when we go back and look at the structure of DNA again, what we know is that it's got this double helix model, and it's made up of 4 bases which are designated by 4 different letters, A, T, G, and C. So what the DNA code does is it directs the synthesis of a specific RNA molecule, so it transcribes into an RNA, which is then translated into an amino acid, which then links up together to form the protein. So that's what should happen, DNA, RNA, protein. So this is how the gene sequence would look like on the DNA code. So you read it at 3 bases at a time. So you have AAA, GAC, CCC, and so on it goes. And each of these triplet code would code for a specific RNA, which would then code for a specific amino acid. So imagine how that might look. So you've got here AGC over there, that codes for a specific RNA, which then codes for a specific amino acid, which then links up together to form your protein molecule. So that's the basics, that's how every gene in our body functions. Okay? So what about this gene code? Would all alterations in our gene code be pathogenic? No, because we all carry certain variations within our genetic material, and that shouldn't matter because the end product may not be any different. So to try and illustrate that example, I've got the table here. It sounds quite complicated, but basically, if you look at GGC codes for a specific amino acid called glycine. Now, glycine is a very important amino acid in OI because it's almost the backbone of your type 1 collagen, and I've got some more diagrams to show that. But glycine is a very important amino acid, and you can see that here, for example, GGC codes for glycine. But what if that C is replaced by a G? So you have GGG. But as you can see, the end product, the amino acid that's produced from this DNA triplet code, is still glycine. So although there is an alteration in the genetic material, in the genetic code, this doesn't matter because the end product, the amino acid that comes out of it, is still the same. And again, you can see here there's all the genes contain a start codon, a stop codon, and the DNA sequence in between. And there's more than one instruction for the stop codon. So just to illustrate the point that not all variations in our genetic code are disease causing. So bringing it all back to OI now, we know of 2 important genes that are significant in OI. And why? Because over 90% of variations in OI are caused due to these faults within these 2 genes, COL1A1, COL1A2, also sometimes referred to as the type 1 collagen genes. Now interestingly, COL1A1 sits on the long arm of chromosome 17, while COL1A2 sits on the long arm of chromosome 7. So they are cytogenetically, if you remember the chromosome picture, they are in completely different locations, but they carry the same function, which is direct the synthesis of your type 1 collagen. And hence, variations in either of these 2 genes causes the same condition, OI. So what COL1A1 does is it codes for an alpha-1 chain, and what COL1A2 does is codes for an alpha-2 chain. And basically, each of your type 1 collagen molecule is made up of 2 alpha-1 chains and 1 alpha-2 chain that intertwine with each other to form this triple helix model. So going back to collagen production again, you can see that the amino acid sequence that, some of the pictures I originally showed, you have the glycine followed by an XY, so that could be any amino acid, followed by a glycine, followed by XY, followed by a glycine, and so on it goes. So glycine is pretty much your backbone, and it's a small amino acid, so it sits quite nicely in that packaging process. And what you have is this triple helix model that then lays down ultimately in the form of collagen fibrils in the skin, bone, teeth, ears, etc. So when we look down the collagen fibrils, for example in the skin on an electron microscope, what you're supposed to see, and this is in normal skin, you're supposed to have on the longitudinal section these collagen fibrils that are tightly packaged and quite neatly laid down. And then here you can see that, ignore the variations in the color, but they're all uniformly packaged and appear very uniform in shape, and that's how it should be. But what happens if there is a variation that's pathogenic, meaning disease causing? So taking you back to that gene sequence again, as I said, you read it 3 bases at a time, AAA, GAC, CCC, and so on it goes. But what if there is an alteration that changes the meaning of the protein that's produced? See here, I've tried to illustrate it with a very simple example, a triplet code instruction saying the old man saw the cat. But what if there is a base that's added, a base that's lost, a duplication of a base, an inversion, meaning it's inverted itself, the base, or a substitution, a replacement of a base. Each of these alters the meaning of the sentence, meaning that it results in a qualitative defect in the protein that comes out. Or what if the stop codon is introduced quite prematurely within the sentence? What you end up having is a quantitative defect, less amount of the protein that's produced. So this is, in essence, what happens in OI. So as you can see, any variations that are disease-causing in type 1 collagen, so for example, if there was an instruction, an alteration that changed the glycine into a much more bulkier amino acid, into a much more heavier amino acid, then it doesn't sit quite tightly, and hence the packaging doesn't happen normally. So you end up with a defective protein, and that's the problem here. So going back to the electron microscopy picture, how might that appear in patients with connective tissue disorders, including OI? What you end up seeing is this collagen fibrils that are not uniform in shape and are not as tightly packaged as you expect. And also what we've come about to see are these impressive looking structures, what are called collagen flowers or cauliflowers. They're basically an amalgamation of abnormal collagen fibrils that come together. And we see that time and again in patients with OI, but also with other connective tissue disorders. Of course, genetics is never that simple. So as you can imagine, I talked about type 1 collagen production using the COL1A1 and COL1A2 genes. But there are several other processes that happen in the post-collagen translation, modification, and laying down in the bones. And there's several other genes involved in that process. And that's why we've come to know that there's more than the autosomal dominant pattern of inheritance that we knew of. So what are the different patterns of inheritance in OI? The commonest form, so over 90% of patients seem to have the autosomal dominant pattern of inheritance. And this is a situation where you have a vertical transmission, so there's often family history. And both males and females would be equally affected, and it doesn't matter the order of the pregnancy or the sex of the child. The recurrence risk would be as high as 50% or 1 in 2. So to try and illustrate, here I've got an example where a parent carries a faulty copy of the gene and the other copy is normal and the other parent is absolutely fine. So when it comes to them having children, the parent with the, for want of a better word, the faulty copy of the gene has to either pass on their normal copy or the faulty copy. So you end up in a situation where if the parent passes on the faulty copy of the gene, the child has inherited the condition. And if they end up passing their normal copy of the gene, the child's okay. So it's 50-50 or 1 in 2. But as you can imagine, there's always exceptions to that rule, the 50-50 or 1 in 2. Why? Because sometimes there could be new mutations. So the variation, the genetic variant could have started for the first time in the child, and hence the risk wouldn't be as high as 50%. There could appear to be variable penetrance, meaning it would have appeared that the condition has skipped a generation just because the features are much more subtle in one generation. Or there can be variable expression where different members in the same family have different features of the condition. So these are exceptions to the rule in autosomal dominant inheritance. Another important exception to the rule is what's called gonadal mosaicism. And this is a situation where we know for a fact that the mutation or the variant is started for the first time in the child. Both the parents have tested negative for that particular variant. But when it comes to discussing recurrence risk, we say that it's not 0%, because of course it's happened once. But also there is the possibility that the mutation, the genetic variant, is present in a proportion of the sperms or eggs without being present in the rest of the body. And that's what's called gonadal mosaicism. And with OI, unlike some other genetic conditions we know of, we know that the risk is there because we've been able to illustrate that. So of course the risk can be as high as 7%, even if it appears to be that the child is the first member in the family who's got OI. And that risk is much higher than some of the other genetic conditions for gonadal mosaicism. Moving on to the kind of more rarer pattern of inheritance, This is autosomal recessive inheritance. And this is a situation where both parents are carriers of a faulty copy each, but it doesn't affect their own health in any way because it doesn't affect the protein that comes out of them. So they are healthy carriers. And here we have a 1 in 4 recurrence risk for children. So how might that happen? So here we have a situation where both parents have got one faulty copy of the gene each, but they are healthy carriers because it doesn't affect their own health. But the only problem is when it comes to them having children, they can either pass on their normal copy or the faulty copy of the gene. So if both parents end up passing their normal copy of the gene, the child's absolutely fine. If one parent ends up passing their faulty copy of the gene, the child is a healthy carrier like the parent, or the other way around. But of course the problem happens when both parents end up passing their faulty copies of the gene. And because this child's got a double dose, they haven't got any normal functioning copies of the gene, they have the condition. So with autosomal recessive inheritance, we say that for the unaffected siblings, they have a 2 in 3 chance of being a carrier. But of course, for the affected person themselves, the risk would be lower for their children because all their children would be healthy carriers because they'd have to pass on one faulty copy or the other. But of course, the risk of them having children with OI would be less because unless they have a partner who's also a carrier. So in autosomal recessive conditions, the risk would very much depend on population carrier frequency. So for example, you've probably heard of cystic fibrosis, which is a respiratory condition. And we know that that occurs more commonly in our population because the carrier frequency is that bit higher. And also we know that the risk of autosomal recessive conditions are higher in consanguineous families where they come from the same ancestry. So hence the same gene pools are passed down. And some of the newer genes in OI seem to follow the autosomal recessive pattern of inheritance. So this is mainly for the rarer forms and the more severe forms of OI. But autosomal dominant inheritance, the 50% one that I talked about, is still the commonest form in OI. So, having discussed about the genetics in OI and the different patterns of inheritance, the next question is, what do we do in a genetics clinic? What do I actually do? Suppose I try and ascertain the genetic diagnosis and try and clarify the inheritance pattern. It's never straightforward. We try and take a detailed family history, clinical history, examine the child or adult, and then have a look at what might be the different patterns of inheritance. Direct genetic testing, especially because there's more than 2 genes in OI now. Try and explain the diagnosis and prognostic information to the family. Discuss the various options available, including prenatal genetic testing, pre-implantation genetic diagnosis, and refer to appropriate specialists. So what happens if we decide that genetic testing is the best way forward to try and confirm the diagnosis? We of course speak to the family, seek informed consent, and then take a blood sample, which is then sent to our genetics lab. And unlike what you see in sci-fi movies, we don't get results within hours. So we've got to try and direct the genetic testing as to which gene to go for. And at the moment, because of recent advances in genetic technology, we're able to look for more than one gene at the same time. So we extract DNA, ask for specific gene panels at the moment, and then all the testing happens. The process itself probably doesn't take very long. But what takes the longest time is the interpretation of results. And why would that be the case? I suppose it's trying to distinguish all these variations and see if they're neutral polymorphisms, meaning when I showed you the change to the genetic code, whether they alter the amino acid or not. And sometimes it's very clear-cut, in which case we can ignore those variations. Or sometimes we have a clear-cut result where we know that it's definitely disease-causing. So for example, if it was a glycine substitution, or if it's been reported in the literature. But of course, it's never straightforward. There's a lot of grey area in between where we find variations in the genetic code which we are not sure if it's disease-causing or not. And that would require a lot of interpretation. So we may have to do software modeling studies, we may have to do animal modeling studies, or extended family member testing, what's called segregation studies, try and test affected versus unaffected members of the family and see if this genetic variant is tracking with the affected members in the family. So there's a lot of work that happens once a genetic test result is not straightforward. And this is what I suppose takes the most time. So ultimately what you'd expect to see is end up with a result where it's directed to the referring clinician, and here you can see what you're seeing is a positive result, meaning a glycine substitution in COL1A1, which has been reported previously and appears to be the likely cause for this child's OI. And this means we're able to now offer testing for other unaffected members in the family as well to try and clarify their condition, their status. So what might be the reasons for genetic testing? I suppose it could be multifold. It may be to try and clarify an accurate diagnosis, be able to offer testing to extended family, discuss options around pregnancy, prenatal genetic testing, pre-implantation genetic diagnosis, to try and clarify recurrence risk for other family members, to explore various treatments available, and that will increasingly happen as newer and newer treatment modalities become available. To try and seek specific management advice, because it's all about genotype-phenotype correlation, meaning trying to find out what the genotype is and what the specific presentations are, and whether we can study more about this. To try and advance the understanding of the condition, but also to try and see if there are research projects that patients can be enrolled into. So there's multifold reasons why genetic testing would be helpful. So to touch upon some of the ongoing research in Sheffield, so we've got an active bone research group led by Professor Bishop, where we try and doing lots of studies in OI. One of the studies I've recently completed is looking at skin biopsy findings in patients with OI, funded by the NIHR Rare Disease Translational Research Collaboration. And some of the electron microscopy pictures I showed you from early on are from that. Looking at exome sequencing, which is basically looking at all the coding sequence of the DNA to see if we can identify the genetic cause in patients where we've not been able to see any variations in the known genes in OI. And ultimately be able to predict outcomes better, but also look at newer treatment options in families with OI. Because at the moment there is treatment which is very good, but it's probably not perfect. Just a quick note on some of the recent advances in genetics as such. So I suppose you've all heard of the 100,000 Genomes Project. This is a Department of Health initiative to try and sequence genomes on 100,000 patients, including a large proportion of rare diseases. And there's a lot of work going towards it, and we're all being designated as genomic medicine centers to try and recruit families. And what's going to happen is all the data that comes out of it would be made available to a GSIP, which is a Genomic Clinical Interpretation Partnership. We're able to access the data and undertake further research on that data. And so be a part of the musculoskeletal domain, and I'm leading on the pediatric bone fragility subdomain to try and understand OI better and the genetics of it better. So I suppose it's quite exciting, but also quite daunting, because there's a lot of work to do. So in summary, hopefully I've covered all the basic terminology, touched upon the genetics in OI, the different patterns of inheritance we see, some of the ongoing OI research, and a bit on the latest advances. Thank you. Happy to take any questions.